Welcome friends and welcome to my channel Sequencial Biology. In this video we will discuss about structural component of virus, and in previous video we have talked about properties of viruses and also their discovery. Usually viruses are two types, the enveloped virus and naked virus, but each virus contains a capsid and their nucleic acid. Okay, the viruses which contains only capsid and their nucleic acid, and this nucleic acid will be either DNA or RNA, then these viruses are called as naked virus. And but outside this capsid, some viruses possesses an envelope outside this capsid layer, and the viruses which possesses this envelope, they are known as enveloped virus. And viruses, remember, viruses have no plasma membrane and no cell wall. Okay, then some viruses have the envelope outside nucleocapsid. So this is the nucleocapsid in green color. We have drawn the nucleocapsid, and nucleocapsid contains the protein coat or capsid, and inside this capsid the nucleic acid are present, their genetic material are present, and this genetic material will be either DNA or RNA. Okay, then outside this nucleocapsid the envelope are actually present. Okay, the envelope is lipoprotein in nature, so envelope is made up of lipid and protein, and for this reason the envelope is lipoprotein in nature, but lipid in bilayer form like plasma membrane. So there is lipid bilayer. So this is the outside layer, outer layer, and this is inner layer. So lipid is bilayer. Okay, and protein is in glycoprotein form, and these are the glycoprotein. And when the virus reproduce inside host cell, then they pick up the host cell lipid from the host cell plasma membrane. So this envelope is made up of lipid component of host cell plasma membrane. Okay, and in this envelope the glycoprotein are present, and which is coded by viral. Okay, so these glycoprotein are actually formed from the viral genome, but this lipid component are made up of host plasma membrane. This is very important. Then lipid part derived from host cell membrane, and the protein coat is virus coded. Okay, so this lipid bilayer and the glycoprotein are together form the envelope. Protein peplomers are antigenic, and when bind to specific host cell receptor they are called as spike. So this protein or this glycoprotein, the subunit of this glycoprotein is called the peplomers, which is present in envelope. Okay, and when this glycoprotein binds with the host cell receptor, then they are called as spike protein. And you know, in coronavirus or the SARS-CoV-2 there are present a spike protein, and which was also the enveloped virus. Some viruses have more than one kind of peplomers, like influenza viruses possesses hemagglutinin and neuraminidase peplomers. Most viruses are enveloped virus, but some viruses are non-enveloped virus. Now we will see the example of different type of this naked virus. The non-enveloped DNA containing viruses are parvovirus, adenovirus, and the papovirus. This is very important, adenovirus. So adenovirus is a non-enveloped DNA viruses. Non-enveloped RNA viruses is picornavirus, reovirus, hepatitis A and hepatitis E virus, and the calcivirus. Okay, now we will see the structure of capsid. And capsid is protein coat, and this protect the viral genome. And here this is capsid, and each virus contains the nucleic acid, and this nucleic acid is covered by capsid, and the component of or the subunit of this capsid is called as capsomers. Okay, then the protein coat and protect the viral genome. This protein coat and this capsid protect viral genome, and this may be single or double protein shell. Okay, the viruses which are enveloped virus, inside this envelope a protein coat is present, and which is known as capsid. But in case of naked viruses, only capsid is present. Outside of capsid there will be present no envelope. Okay, and this capsid is made up of protein coat, and this protect the viral genome. Then if this is viral genome or nucleic acid, this may be either DNA or RNA, and this is protected by this capsid. And this capsid, when is made up of different type of protein, and these are known as capsomers. The subunit of this capsid is known as capsomers when they are made up of different type of protein. But when this capsid is made up of only one type of protein, then they are known as protomers. Okay, a few viruses have only single type of protein in capsid. These proteins are called as the protomers, but most of the viruses have several chemically distinct protein, and which form their capsid, they are known as capsomers. Okay, then this is differences in between protomers and capsomers. In non-enveloped virus, it initiates a first step of viral replication by attaching to specific receptors on host cell. And in enveloped viruses, we have seen glycoprotein are present in their envelope, and this binds with specific receptors of the host cell. But in case of non-enveloped virus, there are present no spike protein, and then directly this capsid layer binds with receptors of host cell, and these capsid are antigenic and specific for each viruses. Okay, so the complete complex of nucleic acid and capsid is actually called as nucleocapsid. So this nucleic acid and capsid together called as nucleocapsid. An additional covering is also present in some viruses around the capsid, just outside this capsid layer. In some viruses, an additional covering is present, and it is composed of lipoprotein, and such viruses are called as lipovirus. Okay, example of this lipoviruses are myxovirus and the herpes virus. Then the viruses are two types: the naked virus, which contains only the capsid layer, and the enveloped virus, which contains both capsid layer and just outside of this capsid layer the envelope are present. But in some viruses, outside of this capsid layer an additional layer is present, which is lipoprotein in nature, and the virus which contains this additional layer, they are known as lipovirus. Myxovirus and the herpes viruses are example of this lipovirus. Now symmetry. Viruses are different shape. The first is the helical symmetry. Capsomers are arranged in helical manner in the capsid. When these capsomers arranged in helical manner, then this type of viruses show the helical symmetry. The helical symmetrical viruses, the appearance of this helical symmetry can be seen in tobacco mosaic virus, influenza virus, and the mumps virus. Okay, so nucleic acid is actually covered by capsid layer or the protein coat, and each viruses contain only one type of nucleic acid. This may be either DNA or RNA. Most of plant viruses are single-stranded RNA viruses, and they are called as riboviruses. Okay, and usually you know that the nucleic acid are two type. Okay, so nucleic acid are two type, DNA and RNA. Nucleic acid are two type, DNA and RNA. These are two type of nucleic acid, and in every living organism both DNA and RNA are present. But in case of virus, only DNA or RNA may be present. Okay, so DNA are usually double-stranded, but in case of viruses we can see single-stranded DNA. Okay, and RNA is usually single-stranded, but in some viruses we can see double-stranded RNA, and this double-stranded RNA is seen only in case of few viruses, and this single-stranded DNA can be seen only in few viruses. The most of plant viruses are single-stranded RNA viruses, and they are called as riboviruses. And most animal viruses, which causes different diseases of animals, they are called as animal viruses, and they are double-stranded DNA containing viruses, and they are known as deoxyribose viruses. Okay, each virion contain only one molecule of nucleic acid called genome. What is this virion? Virion is actually simple viral particle. Okay, so this simple viral particle which is present outside the host cell, they are known as virion, and the virion contains only the capsid layer. Okay, capsid layer, and inside this capsid layer only the nucleic acid are present. So this simple extracellular phase of virus is known as virion. Then each virion contain only one molecule of nucleic acid called the genome, and their genome consisting of nucleotide pairs. Okay, so 1000 to 250,000 pairs can be found in viral genome. The number of genes per virion ranges from 4 to 8. Okay, so in case of small viruses we can found 4 to 8 genes, and in large viruses we can found 100 to 200 genes. Okay, then this is actually the capsid layer, and in this capsid layer the nucleic acid are present, and the single nucleic acid, that is the double-stranded DNA nucleic acid, when this nucleic acid is double-stranded DNA, then they contain 1000 to 250,000 base pairs. Since this is double-stranded DNA, so obviously base pair can be found in this DNA, and the length of this DNA will be 1000 to 250,000 base pair. Okay, and in this DNA 4 to 200 genes can be found. Now we see the different example of viruses. The single-stranded DNA viruses are parvovirus, adeno-associated satellite virus, fudge, 5x174 bacteriophage, S13, M12, F1 and FD. So phages are those viruses which attack bacteria, and the single-stranded DNA containing phages are 5X174, S13, M20, F1 and FD. And usually single-stranded DNA cannot find in any kinds of organisms. Only in parvovirus, adeno-associated satellite viruses and few phages, their genetic material is single-stranded DNA. Double-stranded DNA is normal DNA, and the animal viruses which possesses double-stranded DNA are papovirus, pox virus, adenovirus, herpes virus and hepatitis B virus. And in plant, the cauliflower mosaic virus contains double-stranded DNA, and the phages which contain the double-stranded DNA are T1, T2, T3, T4, T5, T6, T7, lambda phage and phage PM2. Okay, so these all types of viruses contains double-stranded DNA as their genetic material. Single-stranded RNA containing viruses, usually single-stranded RNA are found in every kind of organisms, so this can also be found in a few viruses. Okay, the animal virus which possesses single-stranded RNA are Togavirus, orthomyxovirus, paramyxovirus, rhabdovirus, poliovirus, influenza virus and the rabies virus. These examples are very important in exam point of view. So this poliovirus, influenza virus and the rabies virus are single-stranded RNA containing virus. And tobacco mosaic virus is also single-stranded RNA containing virus, and Brome mosaic virus is also single-stranded RNA containing virus. And the phages MS2, F2, FR and the QB, these phages are single-stranded RNA containing virus. Double-stranded RNA is unusual type of RNA, and this can be found only in case of viruses. So in case of animal viruses, retrovirus and the reovirus, their genetic material is double-stranded RNA. And in case of plant viruses, the wound tumor virus and the rice dwarf virus, their genetic material is double-stranded RNA. Okay, then these are actually the basic structural component of virus, that is the envelope, and inside this envelope the capsid are present, and inside this capsid the nucleic acid are present. And in some viruses, just outside of this capsid and another lipoprotein layer are present. Viruses which possesses this lipoprotein layer are called as lipoviruses. These are actually the most important component of viruses, and viruses are usually made up of this component. Some viruses are enveloped virus, okay, which is lipid bilayer, and they form this lipid bilayer from host cell plasma membrane. So they get this lipid from host cell plasma membrane, but in this envelope the glycoprotein is present. This is solely viral encoded. Okay, so this lipid bilayer and glycoprotein form the envelope, and inside this envelope the capsid are present. But in some viruses this envelope may not be present, then they are called as naked viruses, and inside this capsid the nucleic acid are present. Okay, so these are three most important structural component of virus. And in next video we will discuss about Baltimore system of classification of virus.